Within an organism, each non-reproductive cell contains all of the DNA of that organism. This DNA encodes all of the proteins made in the organism. But if all cells have all of the DNA, how do different cells do different things? How does a brain cell make only the proteins needed for a brain to function correctly, but not the proteins that are only needed in a muscle? So how does a cell decide which genes it should express and which it should not? The answer: transcription factors. Transcription factors are proteins that scan along the DNA looking for specific DNA sequences to bind. Once they find and bind their specific DNA target, they change shape and can interact with or recruit other proteins. Through this binding and interaction with other proteins, these transcription factors can increase, turn on, or decrease, turn off, the expression of specific genes. The main way they do this is to promote or block the recruitment of RNA polymerase, the enzyme that transcribes DNA into RNA. If a transcription factor promotes the recruitment of RNA polymerase, it is helping to turn that gene on. In this case, RNA polymerase will bind more easily and frequently to the gene to which the transcription factor is associated. This results in an increase in the amount of RNA produced from that gene. However, if a transcription factor blocks the recruitment of RNA polymerase, it is helping to turn that gene off. Therefore, RNA polymerase will not bind as easily or frequently to the associated gene, and there will be little or no RNA produced from that gene in this cell. Since the amount of RNA produced by RNA polymerase is usually related to the amount of protein made, transcription factors help control which genes are expressed in which cells and therefore help cells perform specialized functions.